Okay, so we are back again, and this is the third day of the training program, the 3-day training program that is being organized by Department of Bioinformatics here at University of North Bengal. And this third day, we have two very interesting talks lined up today. And these two talks will be based on the R language. So this R language is a statistical programming language, and this is a very useful language for most of the biologists, those who are dealing with the biological data analysis, and particularly the statistics involved in biological data analysis. So we thought of incorporating this R in our training program, and we have two experts in R today who will teach you the basics of R, doing a lot of different statistical analysis, and they will also teach you how to do the graph generation and all. So the first guest that we have today is Dr. Achal Lama, and Dr. Lama is a MSc in agriculture statistics, and he has done his PhD in agricultural statistics from ICAR and IARI New Delhi. He is currently working as a scientist in division of forecasting and agricultural system modeling in ICAR IASRI Pusa. So we would welcome Dr. Lama, and Dr. Lama has a very extensive research experience. He has more than 20 publications. So now I would request Dr. Lama to take over the screen, and maybe he can present his thing. Okay, Dr. Lama.
Thank you, ma'am. Thank you so much. I thank you for inviting me to this wonderful 3-day online session. And with your permission, I would like to share my screen. I hope the slide is visible now. Hello. So, very good afternoon to all. As ma'am said, today our topic will be on use of R in biological data analysis. I will go through the R programming language and some statistical tools that are used for this data analysis, basically for biological data analysis. So these are the outline of my presentation, a few introduction followed by some graphical representation, and then this measures of central tendency. We will be touching bit on measures of dispersion, and then skewness and kurtosis, measures of skewness and kurtosis. So to begin with, I would like to give you a brief introduction regarding statistics as a whole. So it's a subject which is defined as the science of collection, presentation, analysis, and interpretation of numerical data, as stated by Croxton and Cowden. Now, this statistical science has a huge scope ranging from biology, education, economics, business management, and policy decisions. Today, our focus will be mainly on the biological aspects, the data that we get. And the added advantage that we have nowadays is with that recent development in the field of computers that we have now, fast computing facilities, many programming languages at our hand. R is one among them. So due to all this technology, this statistics has become integral part in decision making process, because now we can handle very complex data systems, and you make use of advanced statistical tool to generate or to make proper decisions in our system that we are dealing with. Now, very brief regarding the role of statistics. Why do we want to have some analysis, statistical analysis for whatever data set we have in our hand? this statistics serve as the language of data because data themselves cannot speak. So this statistical tool in various ways helps the data to speak whatever information they have. That is why the second point is provide scientific way. This statistics help us to provide scientific way to extract and retrieve the information that is hidden inside the data. And finally, very important aspect of statistics is that it helps in providing us the prediction or the future values. Whatever statistical analysis we do, we try to capture the pattern that is present in the data, and we try to forecast or have a scenario or future scenario or predict whatever the system may generate. Okay. But there are few things that should be kept in mind while dealing with any statistical analysis. The first important fact is that statistics cannot do miracles. Okay, we statistics is a tool in it itself which can assist the researchers from various fields of various domains, but it will not do any miracle. And second is it cannot change the process of phenomenon. Fine. We cannot change whatever process it exists naturally, scientifically, that cannot be changed. With these statistical tools, we can interpret them, we can understand them, and we can draw some meaningful conclusion out of them. But we cannot change the process of phenomenon. Sometimes it so happens that in various ways or various places we see that it is said that statistics lie. So, what are the scenarios when it can happen, and we should be careful to avoid such scenario. So, in this slide, I'm trying to give just a brief uh, view that how should when are the conditions when we say that statistics lie. Okay, so there are two criteria. The first criteria is that we should have a data set, and our data set can be two ways. It can be the correct data for the analysis that we are doing, or it cannot be the correct or the wrong data set or the misleading data set that we are having. And in the other hand, we can have correct statistical tool that is being used, or the inappropriate or wrong statistical tool. So whenever we fall in this particular zone, when we fall in this zone, that we have correct data set and correct statistical tool, that is the condition which is desired, and that is the place where we will get correct interpretation or the correct inference regarding the tools or statistical ways that we are dealing with. And whenever we fall in these three zones, okay, these three zones where we are using wrong data set but correct statistical tool, or wrong statistical tool but the correct data, and wrong data set and the on at the same time wrong statistical tool. So these three are the zone where the people say that whatever inference they are getting is misleading, or that time they tend to say that statistics lie. But as a matter of fact, when you use correct statistical tool for the correct data set in hand, statistics will never give you misleading results. So in this presentation, I will try to brief you regarding very few basic technologies of statistics, techniques of statistics, which will help us to derive correct statistical analysis or correct statistical inference for our particular data set in hand. Okay. Before going into any analysis, there are few steps that we generally follow as a researcher. We may be our master's thesis, it may be our PhD thesis, or into your jobs when you are into your field of research. We have some steps to be followed. The first important step for planning of any your experiment or any research that you want to conduct is you have to identify the objectives. Whatever statistical analysis you try to do for your particular experiment or for a particular data set in your hand, at first you need to identify the objectives. Your objectives should be clear in research terminology or in statistical terminology. We say our null hypothesis should be clear. What hypothesis are we challenging or we are trying to test out of the experiment that we are conducting? Once we are clear with our objectives, then we try to generate data to achieve our objectives. Now, this data can be generated either by laboratory or field experiments. In majority of the biological sciences, computational biology, the experiments are laboratory based. So we can have a laboratory based experiment, or even field experiment for trial for varietal trials in field in large farms. We go for that. So in that way we can generate data. The second way of generating data is by survey. That is a very common method in statistics. And third is your primary data collection. You have an experimental plot or you have plotted an experiment. You are going and collecting the data from your field or from your lab or from door-to-door survey. That is a primary data set that we have. And second is the secondary data set. The other way of to generate data is secondary data set, which means data set which has been generated or produced by some other experiments, and you are using them either from some government sources, maybe for your computational biology, NCBI databases, sequences you use, or maybe some from published papers or from published government reports or from some other thesis work that they have generated data set and you're using them second hand. So those are secondary data set. So these are few important aspects from which data can be generated. Once you have defined your objectives, identified your objectives, generated your data set. Based on all this, now you have to use appropriate statistical tool. Whatever tool is appropriate for the experiment to test to fulfill your objective, you have to select those appropriate statistical tools. Once you use those appropriate statistical tools, then you can make inferences and from the results that you have obtained. Okay, this is in short that the process to be followed for conducting any research in for doing some valid statistical analysis. Now, moving further, these are few terminologies that are generally used in statistics for any kind of analysis. The first one is observation. Now, what is an observation? The units on which we measure the data is called observation. Fine. Suppose in this online training program or online webinar, in this webinar we have different participants. You all participants that have joined today. So if I want to record the height of participants, so the observation is height and the units are my participants. Right. Now the next terminology that we come across is population. Now what is a population? It is nothing but the collection of all the units. For example, in this training program, the total number of participants that are there is my population as a whole. It can be 300, 400, 500, 600, any number of that is my population. But generally, if you deal with statistics, we hear very often that population size is very large. We try to draw a sample out of it so that we can it is a sample is drawn from population and we try to analyze those sample and make some valid inferences regarding the population at hand, because many a time the population are very large. So it is important for us to know what is sample. Now sample is nothing but a subset or fraction of the population which is taken randomly, but the very important terminology is representative. Fine. Which means a sample should be a representative, means it should hold all the characteristics of the population. That is why it is representative. It is a representative portion of the population which is taken randomly. Suppose, for example, if I record a height of 10 participants randomly out of the total number of participants in this training program, then that is a sample, and my sample size is 10 because I am taking the height of 10 participants. If I would have taken the height of 20 participants, it would be my sample size would be 20 here. Okay, moving on. Now, very random, very often we hear variable. Now, variable is another important aspect, and generally in statistics, we assign the variable with capital letters, capital X, capital Y, capital Z, and so on. And the values that the variables take, we assign them with small letters. So, small x, small y, small z, and so on. Suppose I'm using the example of height again here. So if x is the variable which records the height of the participants, the values x takes, x1, small x1, x2, x3 are the values that capital X, the height variable, will take. Okay, so values can be 150 centimeter, 55, 60 centimeter, and so on. Depends. Now, another important aspect is the type of variable that we deal with. The one is univariate, when we have single variable of interest, suppose only height, and the other is multivariate, This when we have more than one variable of interest, suppose it is height and weight, age, and so on. In biological data sciences, in biological data analysis, we generally deal with multivariate data sets. Okay, we have many variable of interest together. So that is why these multivariate variables are used in our biological sciences. Now we have classification of variable. One is univariate case when you have one variable of interest, and we have multivariate case when we have more than one variable of interest. And if we have only two variable of interest, it becomes a specific case of bivariate. Okay, so now variables on the type of data they record, on the type of values that they are assigned with, can be of two types. One can be quantitative and the other can be qualitative. Okay. Quantitative means it represents some measurable quantities and can be ordered in logical or in some natural way. Okay, suppose price of vegetables or the height itself. Now again within quantitative variables there are two types. One is discrete and the other one is continuous. These things are very common that you hear from day to day. Discrete means it takes finite number of values which are countable. Those are discrete. Suppose number of tillers per plant or number of fruits per tree. It can be 10, 20, 15, 25. It cannot be in fractions. It will be some whole number which is countable. Now in continuous case, it takes infinite number of values which are uncountable but measurable. Means uncountable and measurable. Suppose example is height of plant, length of leaf, or height of the participant itself. It can be 1.43 meter, 2.52 centimeters. Now in this case, what it depends upon the scale of your measurement. The more the finer of your finer scale, suppose from meters you come down to centimeters, you come down to come down to millimeters, and so on. The more you change your scale, the accuracy of measurement you make it more. If the values goes on increasing, it will be 1.433345 and so on. So it is uncountable. There can be many in fraction. It goes on and on, but it is measured. Okay. So these are the difference between discrete and continuous variable. Now the next class of variables is qualitative variable. This is a type of variable which we generally come across in biological sciences a lot. Now these qualitative variables, it cannot be ordered in logical way or in natural way. For example, performance. We cannot order them. We can just give them a ranking. Say it can be good, it can be average, it can be excellent. Similar hair color, it can be black, brown, white, and so on. So these are the two distinct classification of variables on the type of data that they take. It can be quantitative or qualitative. Fine. Now data sets, the very common when we have large data sets, it comes in group. So we say to be group data set. Now, why this? Because sometimes the original values of data are group or data is available in form of groups. Okay, when we have large data set, we are coming in terms of groupings of data. Suppose for plant height, if we have take these examples where the heights are 154, 55, 51 centimeters, 162, and so on. So it can be grouped. Group 1, where we have a height between 150 to 160 centimeters. Group 2, between 161 to 170 centimeters, and group 3 between 171 to 180 meters. So accordingly, the number of observation that they fall, that is 3, 3, and 2, that is the frequency of the data set that we have. Now what happens here in group data set is that the disadvantage of using group data set is that the original values are lost and we have only the idea about the size of the group. But this grouping of data set or group data set is very useful when you have a very large amount of data set and at one go you want to know the information or the structure. How are the data set or how are the different points in the data set grouped or how they look like. These at those juncture, these group data sets are very important. Now the next is classification of data. Now this is very important in biological sciences because classification means process of arranging data into groups or classes based on similarity and resemblance. Fine, because based on their similarity or their resemblance means based on their how similar are they or how together they can be grouped. So this is the classification of data. Okay. Now there are many, as because we deal with multivariate data set most of the time in biological sciences. There are many, statistical tool like cluster analysis and all. So this is beyond the scope of this lecture. But the classification of data is very important because at that time we can classify based on the resemblance and all. Okay. Now moving on, when we have group data set or we have classified the data, we have or based on this, we have formed groups of the data based on their similarity or resemblance, then we say it to be classification. So once we have a data set, group data set, we have classes of the data set, and then we try to define the frequency of it. Okay. So when we say frequency, means what are the number of observations that are falling in that particular class. So there are two terminology. One is absolute frequency, and the other one is relative frequency. So in pass and fail, when we have absolute frequency means the number of appearance of pass. So it is A is equal to 7 if A is termed to be the number of students that have passed in a particular course, and F is number of students that have failed. So B is equal to 3. So the absolute frequency are 7 and 3, but the relative frequency are 7 by 10. The total number of frequency that is 0.7 or 70%, and it is 3 by 10, 0.3 or 30%. Okay. So this is the R code that I used for getting my absolute frequency and relative frequency. In the end of the class, I will give you some brief outlook regarding all those codes. Uh, Similarly, where you can name the data, enter data as X, you can change the name of the data as per your convenience, and P as 1, and you can mark them, and F as fail as 2. Similarly, you can put them in a enter them in the variable result. Okay, and then finally you can table them and you can get absolute and as well as relative frequency. Now, the another important aspect is frequency distribution, because when we have uh, data set, ungroup data set, uh, here we can arrange them into a frequency distribution, because until and unless you have you don't have, if you have a frequency distribution of the particular data set, many properties like mean, median, mode, and skewness, curves, all these properties you can visualize. And once you can visualize these properties, it becomes very easy for you to understand the different characteristics of the data and to draw some meaningful conclusions. So first we have to divide them into entire range into classes, as I said earlier, based on their similarity, okay, or resemblance, and each class has a lower and upper boundary of a class, which is said to be the lower limit or the upper limit respectively, and the difference between the limit is called the width of the class. The difference between the upper limit and the lower limit is called the width of the class or the class interval, and the value of the variate, whatever value that we take, it lies between the lower and the upper limits. Okay. So this is an example how the class interval is between 10 to 15, and this mid value is upper plus lower, divided by 2, that is 12.5. Similarly, for the class 15 to 20, the mid value is 17.5. So it is said that all the values will revolve around this particular value, mid value. Okay. So the number of observation in a particular class is said to be absolute frequency or the frequency, or in general term the frequency. And when the frequency is divided by the total frequency, it is said to be the relative frequency for that particular class that we have discussed earlier. Now the new concept is, the new terminology here is the cumulative frequency of a class, which means the number of observations less than or equal to upper limit of a particular class. Okay. In further slides, it will be more clear to you. Now, to understand all this, whatever I have said in previous slide, now let us take a hypothetical example of a number of fruits per tree in a mango orchard. These are the variable I have taken NF, number of fruits per tree in a mango orchard. I have given it to the NF, which is assigned with all these values. So I can have a range to understand its upper and the lower limit. So when I type range NF, it gives me the values 32 and 84, and then I can, based on this range, I can decide the class with class. So class will range between 31 to 84, so 90. So class width is being 10, I'm taking 10 as class width, and the sequence from 30 to 90. So if I break them in R, I can create classes. So break them, so I have to write sequence between 30 to 90 by 10. So if my class would be from 20 to 100, here it would be 20 to 100, and width is 20, then by 20. So when I write this particular code, I get such output 30, 40, 50, 60, and so on until 90. Okay. So I can cut them into different classes and using this cut function. And when I write table.cutnf, I get the absolute frequency of different classes. It is 5 for 30 to 40, 3 for 40 to 50, and 3 for 50 to 60, 5 again for 60 to 70, and 2 for 70 to 80, and 2 for 80 to 90. This is how I get. And if I want to see them in tabular form, what I have to do is I have to just write the code cbind, that is column bind, cbind. Just I have to type cbind and the rest code remains the same, and then I can get a tabular view. So you can see the tabular view out here. Fine. So this is how it is achieved. Similarly, for relative frequency, it is table divided by length. The length means the total number of frequencies. So I get a relative frequency automatically. I need not to bother. And similarly, if I want to have a tabular view, I can write cbind, and I have stored this as RF, means the relative frequency. Okay, so this is how I get the relative frequency as well. Now, as I was talking regarding the cumulative frequency, from cumulative frequency we get the cumulative distribution function. Here we can use cum sum means cumulative sum up to that particular point of time, and here is the cumulative sum. So we have 20 number of observations in total, and for the first class the number of observations is 5, up to second class the number of observations is 8, similarly 11, 16, 18, and for the last class the total number of observations 20. So it keeps on adding the number of observations. If you minus it, 8 minus 5, you get 3, which means there are 3 number of observations that are falling in this particular class. Similarly, if you subtract it with the previous class, you can understand what number of observations are falling in that particular class. Or if you subtract between this 18 and 5, you can understand how many observations are falling between the class 30 to 40 and 70 to 80. Okay. Similarly, if you want to have relative frequency, you can come sum table divided by length. You can easily get a relative frequency. So if you want to understand this is like up to class 60 to 70. If you look here, 80%, 0.8. So 80% of the observation lie within the class 60 to 70, from 30 to 40. Okay. So this is how you can interpret them. And if you want to plot them, you can use this particular code plot, which I'll explain while I will be doing my practical in hours. Okay. For graphical representation, if you want to plot, you can. This is the simple bar chart that I'm using it. Now, the important in bar chart is that it makes bar of equal width. These widths are equal, but these heights are different. If you look, now these heights are different. Okay, this is different from this particular height. Now, this height depends upon the magnitude of a quantity. Okay, this height depends upon the magnitude of the quantity. The more the value, higher the height. Okay, less the value, less the height, but the width remains constant. Fine. So this all depends. So this is a simple bar chart that you can have, and you can understand the different values that are there. With respect to height, you can understand height of the bar. With respect to height of the bar, you can understand its the values that it is taking, whether it has more value or it's taking less value. In R, we have simple code for as bar plot, where we can get, and the name fruits per plant you can change here. X lab is labeling your x-axis. You have given trees. You can give your the name as per your convenience. Y lab is labeling your y-axis. Given number of fruits. Here it is number of fruits. So it totally depends upon what type of bar chart you are getting, and what x-axis, y-axis you want to give, and what name you want to give for the chart. You have that flexibility in R. You can simply write them within the inverted commas, and it will get displayed in your bar chart. Fine. Now let us take another example where I will be trying to explain you how to take a matrix data input into your R. So this is a matrix. Okay. Here we have three number of columns for each plant 1, plant 2, and plant 3, and we have 4 rows for each day 1, day 2, day 3. Now, this is a hypothetical data from experimental field. It will be filled spelling mistake, sorry for that. And what we are trying to do is we are trying to record the number of insects per leaf in each plant, plant 1, plant 2, and plant 3 for each day, day 1, day 2, and day 3, and day 4. Fine. Now, if I want to input this into R, I have this table made in Excel. Suppose I want to input this into R, so what I have to do is I have to write matrix. I have to define the number of rows, number of columns that I am trying to make out of the data set, the number of the dimension of the matrix I have to assign, and then I have to assign the data set using data is equal to C, and then by row. I'm doing it by row. You can do it by column also. By row is equal to true means it will take input as row. So you can see 542 hello of my first row 542, then 654 hello of my second row 654. Similarly 524 hello of my last row 524. So once I do it in this way, enter the data set in this way. R will take it as a matrix with number of rows 4 and number of columns 3. And if you want to visualize it, you have to just write the name of the matrix that I've saved is NIL, number of insects per leaf. If you just write this and run enter in R console, which I will be showing it later, you will get this particular matrix. If you match them, it is the same matrix that I wanted to show. Okay. Now, so if the same matrix you want to, you want to have stacked, you want to have stacked bar chart, and then you can have in this way. So each blue, green, red, and yellow, these are the representation for number of days: day 1, day 2, day 3, day 4. Here I have used these colors. If you want, you can change the colors. You have an option of changing the color combination in R. This is I have taken it randomly, so you can understand the number of in day 1 what are the number of insects in plant number 1. These are the plants. In day 2 what are the day 3, day 4. So you can easily visualize and compare among them. So this type of visualization is easy. Visualization will help you easily compare among the plants with the number of insects per day in daily basis. Okay, as you can see here, it is least number of insects blue the first day for plant number 3. Similarly, you can understand this. Okay, you can have easy visualization. And another way of showing your stacked bar plot. The third one and the other one is your pie diagram. This is a very common diagram that has been used. Now here the area depends upon the, as we have 360 degrees in total for a circle. So definitely the angle is determined by the relative frequency. Okay. So if I take an example of, suppose I'm taking an example that we have 150 participants. Out of them 80 has master degrees, hold master degree, and 70 is hold, and 70 of you all hold PhD degree. So if I am to plot them using pie chart, so this is how I get, because as the master degree is more, so you can see the area under master degree, the blue area is more as compared to my green area for PhD. So similarly, if we have pi as a command in R, and we can give the labels, master degree, PhD. If you have more labels, you can of course keep on adding the label. What you want to give the name, I said qualification of the participants. So main qualification of the participants within inverted comma, whatever you give, it will happen. And the color I have given blue and green. Of course, there is again flexibility for changing the color that you want to put on. Okay. Now moving into the another important statistical tool for having understanding the data set in hand, that is the measures of central tendency. We have many measures we will discuss briefly due to shortage of time. So when actually this measures of central tendency comes into picture when you have numerous variables in a data set and each variable has many observations. So when we have dealing with multivariate case, multivariate variable, more than one variable of interest, and each variable has more than one observation, many observations. So it is on those scenarios, it is very difficult to dig out information from each observations. It is very difficult. for each observation, for each variable, you try to find out some information. It becomes very difficult. So in those cases, you want to have summary. What we are interested as user, or you are interested as user, is to have a brief summary of information that is hidden inside the data. And this brief summary is generally given up through the different tools of under measures of central tendency. Because these are very important, because we generally conceive our information in some sort of averages. Suppose, as I give an example, is that average performance of student is 80%, which means we give an idea that, suppose for example, the average campus recruitment package of one particular engineering college, say X, is 5.5 lakhs per annum, which means this is an average value. So given on the average value, we think we try to rank our preference for selecting a particular college. Fine. So we generally conceive our notions in all average values. So in statistics, these average values are referred to as central tendency of the data. These are say point towards which the data tries to accumulate or move. Okay. So these are the different measures: arithmetic mean, harmonic mean, geometric mean, median, mode, and quantiles. We will briefly touch all. So in case of arithmetic mean, this is the example. This is the equation. You will get it in all standard textbooks, and in R it is very easy. You just need to type the mean and the value of the variable number of fruits. You will definitely get the mean. But the problem is, if you have very large data set and you have some missing observations, you are not sure that your data set may or may not contain some missing observations. So I have given an example here that I have some undefined values or some missing values. So in that case, I have to write an extra line: na.rm is equal to true. So if I give this particular line along with mean nf, then this missing observation values will also be taken into account, and accordingly you will get the mean of the observation. So when we deal with large data sets and you're trying to calculate the mean, this particular code is very useful. Okay. So this is arithmetic mean for group data. I'm not going into details, but what is important is you can also have weighted mean. Weights wi can be assigned accordingly for giving means. So this weighted mean concept is sometimes very handy while dealing with some biological data, because for each data set you try to give predefined weights. So in those cases, this weighted mean concept is very much helpful. And this we have a weighted dot mean function inbuilt in R, where you need to give the frequency, those corresponding frequency, and those corresponding median of the classes, the middle value of the classes, and it will give you automatically define you, return to you the weighted mean values. Okay. And the next important thing that we use, or statistics we use for central tendency, is median. Median is generally it is a positional average only. It will give you the position, the middlemost value. Okay, the middlemost value. And now, if you have odd number of observation, it is very easy to calculate. The median is the value of n plus 1. N is the number of observation that you have had in hand. N plus 1 divided by 2. Suppose you have 7 number of observation. So 7 plus 1 is 8 divided by 2 is 4th. So your fourth observation is your median. And you have even number of observation, then you have n by 2, and n by the middle, the if you have 10 number of observations, suppose even number of observation, the fifth observation and the sixth observations. Okay, fifth and the sixth, the average of fifth and sixth observation will be your median value, right? If you have even number of observations. Similarly, for if you have group frequency, then this is the formula. It is available in your standard textbooks, and in R, when you have group frequency, still the previous formula will work for you. Now, another important measure of central tendency is your partition values. Uh, this is very important for us, because when you have large number of data set and you want to see that below what particular value is my 25% data set lying, or my 70% data set lying, or my 80% data set lying, or my 90% data set lying. So these cutoff values are very important when you are in biological sciences, when you want to understand the various relationships. So in in terms of statistics, we call them partition values. Now, the important partition value that we generally deal is median. Median means which divides the total data set into two equal parts uh, and two equal halves. So that is median. Quartile, which divides the data set into four equal halves, where Q1, Q2, Q3, and so on. In decile, which divides the total data set into four equal parts, and in percentile, where it divides the data set into 100 equal parts. Okay, and so on. In R, we have a function called quantile. Okay, we say it to be quantile by default. By default, this quantile takes divides the data set into my four different values between 0 to 25%, Q1 25 to 50%, Q2 50 to 75% is Q3. If you can see here also, Q1 this median is equal to Q2 positional average. Median is equal to always the second quartile, and Q4 is between these two observations. So you can see it divides into four equal parts. Between minimum value to this is Q1, this is again my second part, this is my third part, third equal portion, and this is my fourth equal portion. Fine. So 0% is the lowest value and 100% is the highest value for all partition that we do. Now, as I said, when if you want to divide your data set into 10 equal parts, it is said to be decile. So what extra we have to do is we have to define a sequence. Okay, sequence. So the range of probabilities between 0 to 1. So between 0 to 1, if you want to have decile, you need to have an increment of 0.1. Okay, so you will have 10 equal parts like this: 0.1, 0.2, 0.3, and so on till 1. Okay, so if you do that, the data set in quantile data set, comma, the probability value 10%, 20%, 30%, 40, 60, 70, 80, 90, and then 100. Suppose you are interested to see uh, what is the value up to which value the 80% of my data set lie. So 80% means it is 68.8. So this is the value below which the 80% of the data set lie. Means if you sort your data with less than equal to 68.8, value of 68.8, you can extract 80% of your data set. Suppose you want to extract 90% of your data set, then you have to use your sorting. The upper value of sorting is 74.9. If you use your upper value of 74.9, which means less than equal to 74.9, the 90% of your data set will be separated. Fine. Suppose you want to have more exact values like 92%, 77%, 75%, 42%, 43%, more exact probability values. Then you have to divide the sequence into 100 equal parts. So it will be between 0 to 1 and with a sequence value of 0.01. Okay, so these are the 100 probabilities that you get these values. So randomly 53%, 0.53, 86%, 0.86. So depending upon your requirement of your analysis that you want to do or you want to present your data, similarly way you can decide the partition value that you have to use, and R gives you all flexibility to choose any amount of value. So if you want more precision, you can increase it from 0.01 to 0.001, or if you want at 5 at zero point 0.5 interval, you can make it 0.05. It totally depends upon you. R gives you all flexibility to partition your data at whatever precision level you want to partition it. Okay. Now the next important word is mode, and mode is the value which occurs most frequently in a given set of observation. That is the value of a variable which is predominant in the given set of observation. Okay, so this is mode for for group data, frequency data. These are available in standard textbooks. I'm not going much into details of it. Uh, so this is all for mode. So mode is basically the value which occurs the maximum time in your particular data set, that is said to be mode. Okay. Now when we have mode, when we have median, when we have mean, all this, what is the requirement that we need to go for measures of dispersion? Okay. Now the requirement is, you can understand from this particular example where x has takes the value of 10, 15, 20, 35, and 30. Its mean is 20. Again, if you take another variable y, which takes value 2, 8, 10, 20, 60. Again, its mean is 20. So distinctly you can understand this variable x and variable y, though they may have same number of mean, same value for a central tendency mean, but their distribution, but their values, there is a huge difference between the two values of the values of x and values of y. To capture such disturbance or such deviation, such deviation within the data set, the measures of central tendency is not adequate, so that is why we move towards measures of dispersion, where where we can understand or capture some more qualities or some more characteristics of the data set. Among the measures of dispersion, we have basically two important measures. One is absolute measure of dispersion, and the second one is the relative measure of dispersion. Among the absolute measures of dispersion, we have the range, which is the lowest and the highest value, and then we have quartile deviation or semi-interquartile range. We have mean deviation, and we have variance and standard deviation. So in our day-to-day life, we generally tend using range for biological sciences, and then variance and standard deviation is used a lot. Sometimes, in some very specific cases, mean deviation is used, and very rarest of the cases, we generally try to tend with quartile deviation or semi-interquartile range. So these two things, range and variance and standard deviation, is very important for us. And among the relative measures of dispersion, we generally go with coefficient of variation. Others are also there, but we generally we use coefficient of variation. So we will see what are those, what are their formula, and how they are calculated are. Okay. So range, as I am already saying, it is the difference between the maximum and the minimum value of given observation. So maximum xm denoted by xm and xo is denoted by the minimum value. So you simply type max of nf, the data set name of the data set, minus mean of nf, you get the range. And in R there is a default command range nf. It will just give you the lower value and the upper value. Okay. So if you want the exact the lower value and the upper value, thus the values, you type the command range. And if you want the difference between the lower value and the upper value, how much dispersed is the data set, then you can use max of nf minus mean of nf, and you can print them. So it is 52. So depending upon your requirement, you can use the command. Okay. Similarly, for quartile division or interquartile range, we have inbuilt function of IQR. So you write IQR, and if you want quartile division, it is IQR divided by 2. So you get the quartile division. Okay, I'm not going into details of this much. So similarly, for mean deviation, we generally do not have an inbuilt command, but we can write it down. So the mean of my variable, the main variable minus mean of my variable, absolute value, because it is absolute, means the negative and the positive, means the sign is ignored. When we take the absolute value, the sign is ignored. So it does not give you the direction. When we ignore the sign, it does not give you the direction, because it is either positive or negative. It will just give you the magnitude. What is the difference? But this is one of the disadvantages for using mean deviation. Okay, when we take the mean, we get the mean deviation. Similarly, the coefficient of mean deviation is mean deviation divided by m is the arithmetic mean, because we are taking it from the arithmetic mean. So you will get this mean deviation. Now coming into the important the portion of measure of dispersion, that is population standard deviation or the sample standard deviation. We sometimes say the sample, we generally call it standard error also. So these are the standard formula when the value is deviated from its mean value squared, summed over, and the mean of it is taken and square root of it. So this is always a positive value. And similarly, for a standard deviation for sample, it is n minus 1. I'll not go into much of the details, because 1 degree of freedom n minus 1, because we need to estimate sample mean x bar. Instead of you are using x bar, the sample mean instead of the population mean mu. So while estimating x bar, we use 1 degree of freedom. So it is n minus 1 over here. So the formula goes like this. Okay. In R, we have inbuilt, you can simply write variance of nf, bhag of nf, so you get the variance, and of course square root of variance is your standard deviation, you get ht. In R, It calculates on basis of sample. So you need not to worry. You will get sample statistics. That is sample deviation you will get, or you can say the standard error you will get. Whatever HD is there, which is the standard error. So coefficient of variation, which is very much used in our day-to-day life, it is nothing but sigma divided by x bar, means the mean into 100. Okay, and it is written in percentage, and it is a pure number. The unit of observation cannot be mentioned with its value. It is unitless. It is a pure number, and it is given in written in percentage. Okay, and this is how you get standard deviation by mean. We have calculated standard deviation already, and CV is CSD into 100. You get the CV value, and you get it to 30.5. Okay, and in R we have an inbuilt function called summary. Summary of, when you use this inbuilt function summary, we automatically get this many values. We get the minimum value, we get the first quartile, median means the second quartile, and then we get the mean value. Mean means the arithmetic mean. We get third quartile, and we get maximum value. Okay. Now, if in brief, if you want to have just these values or brief outlook, you can simply type summary and the name of the data set that you want to have, you will get these values. But for specific values, if you want to have specific values, so I have shown you different codes. Now, another very important aspect for understanding the type of data set, or any outlier observation is present in a data set or not, or you can compare different data set with the help of box plot. Okay, and box plot, you definitely you will get these observations: the minimum value, the maximum value, the first quartile, the third quartile, the median value, and of course, if there is any outlier present in the data set, it marks it. Okay, and the difference between the third quartile and the first quartile is my interquartile range, which we have discussed earlier. So this is a representation of my data set NF. This is the original data set NF. So this is the block plot. You can see the minimum value, the maximum value, the Q1, this is Q3, and this is my median. Fine. But here, if you look, I have introduced four values, 1 and 5, and 150 and 170 values in intentionally as outlier values. So when I have these values, box plot NF1, so you can see these two values, 150 and 170. It is showing as my outlier, and you can definitely see the values have the minimum and the maximum values have also changed, because the scale here is from 30 and scale here is from 0. Fine. So when the values change, the minimum maximum value change, and according to minimum and maximum value, it is showing me that the value 150 and 170 is my outlier. So at one go, I can have a look that whether any outlier is present or not, and also other statistics: what is my minimum value, what is my maximum value, and what is my where do my median lie, where is the middlemost value situated. Right? This will help us. This helps. This is a one way help from box plot, why you can compare two particular data set, a data set where no outlier is present and a data set where outlier is present. Okay, and the other aspect is skewness, which means lack of symmetry. Many a times in our biological data analysis, we come across this phenomenon of skewness, where symmetry lacks. Means it is the data set, as you have heard regarding the normal distribution. Okay, now this is the symmetry. The skewness totally depends upon the shape of curve obtained by frequency distribution, and it generally depicts the nature and concentration of observation towards higher or lower values of area. Okay, we can have a negative, you know, negative in negative direction. We can also have in positive direction, and this the mean, median, and mode. The position of mean, median, and mode in particular frequency distribution defines the symmetry. Now, if my mean, median, and mode all are present in one particular point, then it is said to have no skewness, or it is said to be a normal curve. And if my mean is positioned further from mode, and median will always be in the middle, so then it is my positive skewed. And but if my mode is positioned further away from mean, then it is said to be my negatively skewed. This is how the graph looks. Okay, for skewness and positively skewed, negatively skewed, and for positively skewed. This type of plot you will get. Okay, and the value skewness coefficient in R by default under the library moments we have the command skewness. If you give the value skewness and the name of the variable, the data set that is stored NF, you get the value of skewness. Okay, now, if it is equal to 0, it is symmetric. If it is greater than 0, it is positively skewed. Less than 0, it is negatively skewed. So, for the first data set, NF, the original data set number of fruits, a value of skewness 0.057 is very close to 0. So, we can somewhat say it to be symmetric. But for the second case, the value is 1.1346, which is higher as compared to much distinctly away from 0, so we will say to be positively skewed, and this is due to introduction of the outlier values 150 and 170 into the data set. Okay, now the next concept is my kurtosis, which gives me the pickedness or flatness of a curve. Now this is the normal curve, the middle curve is the normal curve, you can see. Okay, if my curve is picked, okay, picked in this way, then it is said to be leptokurtic. If my curve has flattened this way, it is said to be platykurtic, which means if my data sets are distributed, if my variance in the data set is more, then we say to be platykurtic, and if the variance in the data set is less, okay, we say to be leptokurtic. Here the kurtosis coefficient is given by B2, B2, beta 2, and here if value is 3, it is said to be mesokurtic or normal. Greater than 3, it is leptokurtic. Less than 3 is it platykurtic. Fine. And the term is kurtosis, and the command is kurtosis we use, and the value for the original data set number of fruits is 1.701, so which is less than 3, which is platykurtic. But for the value NF1, where 150 and 170 were the two data set, the 2 points as outliers I have used, and for those 2 points, this value has become 5.36, means it is leptokurtic more than 3, and this is platykurtic less than 3. Fine. So you can see that 2 points, two outlying points, if present in a data set, it can change the the characteristics and the nature of data set to such extent, the shape of the curve to such extent. That is why work plot comes very handy to understand is there any presence of any data set, any outlier in a particular data set or not. Okay. Now, before closing my presentation, the theoretical part, I would like just to read these lines and just a word of caution that though we have many statistical software has made sophisticated analysis, most sophisticated analysis feasible, problem arises when the user applies them without understanding the assumptions and limitations of the procedures that we are using. So I hope this 2.5 day till date and one more lecture to come will has enriched your knowledge to select the appropriate tools, statistical tool or any computational tool for your particular data set and use of the various software that has been exposed to you all, and so that you can make appropriate decisions. Right? So these are the references that I used. You can follow them, and this is thank you for the theory portion. Now I will like to share with you all my uh, I would like to share uh, some code, uh, okay, R code that I have shown. So I hope this window is visible. And
Yes, just a moment, Dr. Lama. Yes, it's now
yes, yes.
visible.
Okay, okay. So these are the R codes that I have shown you, of course, in the course of my slides also. So this is the dataset number of leaps that I had been taking you. In R, basically, we have windows. This is the my R editor window where I write all the commands. Okay, this is my console where my commands will run. If there is some problem in the command that I'm using, it will give me a red sign symbol and it will show. It will tell me what is the problem. There is a problem in the common, and I need to correct it accordingly. Okay, now this is an global environment window where whatever variable I will run, it will be stored. The name of the variables will be stored, and the type of variable will be stored here. And here in this console, in this particular console, you can see there are many tabs, files, different files that are there in R. Plots, any plot that I have plotted here will be coming here that we will see, and most importantly packages. Now R is a free soft freeware, you all know, and it comes with some packages, inbuilt packages in R, and many other statistical packages that are built for particular analysis. So accordingly, if you want to for your particular analysis, you can download any package from R CRAN, the repository for packages. If you type in Google, you will get it. You can download, and after downloading it in a system, you have to install it. Okay, you have to install it like this. So if anybody is interested regarding all this procedure, you can drop me a mail. I will share with you the appropriate steps with you. PDF is there with me. I can share with you all so that you can do it. But for today class, limitation of time, I'm already running a bit late, so I'll be very quick. So this is the NF, number of fruits, and so once I run it, you can see this has popped up here in this particular environment. NF, it is a numeric value taking 1 to 20. There are 20 number of values. So if you want to see, and you can see there is a blue line here in console, which means it has run properly. Okay. So NF is the number of fruits. You can see it has run, and this is the values. So if I am to go for range, so it is range NF. So it is giving me the range 32 and 84 is the range. Similarly, for tracks, if you want to make classes and you want to cut them into classes, as I have shown in you in my slides, you can see these are the classes labels 30 to 40, 40 to 50, and so on till 80 to 90. This is how I can make classes, the group frequency. Now if I table them, so if this code table will give me what? Will give me the absolute frequency 5, 3, 3, 5, 2, and 2. So if you want to delete this window, clear this window, you have to do Control L. You have to type Control L, this console window will be clear. Fine. And as the number of variables I am running, you can see here all the names of the variables are being stacked in here. Okay. So similarly, table, then C bind, column bind, you will get this in tabular form. For relative frequency, you can again, so column bind, you get the relative frequency 0.25, 0.15, and so on. And if you want to further, if you want to plot them, so this is the plot I was talking about. When you plot, the plot will come here. So this is the cumulative frequency plot. If you remember my slides, I have shown you this particular plot in my slide. And as the number of variables are increasing, the more and more variables, you can see here also the number of variables are increasing in this environment window. Okay. Similarly, for bar plot, if you use the bar plot that I have shown, so you can see here, this is the bar plot. The code is simple, bar plot, and so on. And for matrix, as I was talking about, you can have the input of the matrix, and if you look into the matrix, so I'll just clear this console and I'll run it once more. See the matrix. I hope you can see this matrix. See, this is the matrix that I was talking about, and here also the matrix has come. The name of the matrix data has come. So the dimension 1:4, 1:, number of rows and number of columns are here. So stacked bar plot also code is here. You can see again the line graph. Also you can see this is how the line graph comes. For pie chart, number of participants that I was talking regarding that. Also you can see the pie chart. It is coming to you. And for mean values also you can see the mean values are coming. Similarly, all the codes that I have shown you are given here, and it runs one by one. I will be sharing this code with the organizers, so you all can run it. So I hope I should finish it here. because I have just exceeded my given time limit. So I think I'll stop.
Okay, thank you, Dr. Lama, for that
Thank you,
very
madam.
informative session.
I was bit, it was bit in hurry, so I could not complete all. But I
No, no, no,
assure
no.
that I will be providing all
Yes,
the
yes,
code, and
yes.
so if anybody is interested, then they can of course.
Thank you, Dr. Lama. Now, for the participants, if you have any question, please comment them in the comment section of the YouTube so that we can have a question answer session. Meanwhile, I'd like to add one thing just that before the starting of this technical program, we have told you to install the R program, but we have forgot to mention the R Studio. So Dr. Lama has just shown you to run some of the codes in R Studio. So this is just a suggestion to all of you. After you have installed the R codes, you can install the R Studio in your computers. R Studio is also freely available. You can search the Google with the R Studio, and that will install the R Studio. And R Studio has this graphical user interface, so you can use that R Studio for plotting and running the codes as well. Just a suggestion, so you can search the R Studio for that. Meanwhile, there is a question, Dr. Lama, I would like
Yes,
you
ma'am.
to,
Uh,
yes, how to plot the heat map. This is a very separate topic, but you can just
yes, of
answer
course,
me
of course, of course.
something.
Yeah, actually, heat map are generally generated when we try to understand the correlation relationship between two variables. This was what I have not covered. I think the next speaker will cover. If he does not cover, I will definitely share the code with ma'am, so ma'am can of course make you one. Okay.
Okay, thank you,
It is
Dr. Lama.
available in R. It is available in
Yes.
R. So I will definitely share it. I will write
Yes,
it and share it.
yeah, sure. So we'd like to add over here that as Dr. Lama has told us that we will be sharing the codes with you. I think he will provide us the code, and at the end of this training program, so we'll share the codes with you. So if you want to rerun the codes in the R Studio, you can do that. So I think that will be a very good idea to do that. So we'll share the codes. Okay, in the meantime, if there is any another question, I think the same one has been suggested by another thing, the R code. Yes, we'll do that. Just as we have mentioned that we'll arranges the code for you so that you can run the codes. Acha, meanwhile, I can't find any other question. It's all about only comments. Thank you. So I think, yes, thank you, Dr. Lama, once
Thank you,
again
ma'am.
for,
Thank you so much.
yes. So thank you for joining with us. And we hope to maybe in the future also, if you arrange some other thing. Acha, last one, there is a comment. Okay,
Yes, miss.
please comment on looping in art. Okay, so can you
Okay,
take that?
okay, okay.
Yes.
Yes, ma'am. Actually, whatever other programming language you have, if loop, for loop, if else, if then, whatever loop structure is present in another other programming language, in object-oriented programming language, similar looping structure is also present in R. So, if you are interested, I can, share syntax for loop and if loop, which are commonly used. So, I will share that also along with this particular code, so you can of course try it out yourself.
Okay. So maybe Dr. Lama, you can give your email ID
Yes,
over.
ma'am. Yes, ma'am.
Yes, sir. So if any of you are
Yes, ma'am. Yes, ma'am.
interested, so we'll provide you the email ID of Dr. Lama. So
Yes,
you
ma'am.
can directly contact him with your queries, or maybe if
Of
you
course.
have any other questions, so you can contact him.
Yes,
So
ma'am.
Dr. Lama, thank you. Thank you. We
Thank you
so
so
want
much,
to know.
ma'am. I would
Yes.
like to place on record. Thank you, and I do it was been wonderful talking to you all, ma'am, with the and giving this presentation. I thank you, Department of Bioinformatics, and Chiranjeev Sarkar also for inviting me, ma'am. Thank you so much. It was nice.
Okay, thank you, Dr. Lama. So we'll move to the next speaker now. And our next speaker is, yes, our next speaker is Dr. Rahul Banerjee. And Dr. Rahul Banerjee has done his MSc in Agriculture Statistics from ICAR, ARI, New Delhi. He's currently working as a scientist in the Division of Sample Survey, ICAR, IASRI, New Delhi, and he has more than 10 international publications in reputed journal. So Dr. Banerjee, welcome. I welcome you on the behalf of University of North Bengal Department of Bioinformatics. So I would like
Thank you.
you to take over the presentation, so you can start your presentation.
Thank you very much, ma'am, and a very good evening to everyone. So let me share my screen.
Yes, sir, it's visible. You can just make a full screen. It's visible. Okay.
Is it visible now?
Yes, it is.
Okay. So the topic of today is statistical analysis of biological data. Now, this is a very wide topic because we know that there are lots and lots of statistical analysis which can be done on biological data, and there are different, depending on the data, depending on the problem, there are lots of analysis. So here we'll be basically focusing on some of the inferential aspects of the biological data, and also some of the plotting aspect of how we can plot the data. So the first question. Okay. So the first question that comes to our mind is that why do we need for analysis of data? As we can see that there are basically three steps. First we have a data, and then we have analysis, and then we have idea. Whenever we perform an experiment, whenever we perform an analysis, or we perform any kind of a survey, the first thing that we get is data. Now the data are crude observations. It can be numbers, it can be alphabets, it can be anything. So the important thing is that we have to extract information from this data. And that process of abstracting information from the data is known as analysis. So this is a three-step process where we have a data, and then we do certain analysis, and then we come out with an idea, or we come out with the information that is contained, or that is basically hidden in the data. So here we can see that to answer research questions and determine the trends and find out the relationship among the variables is why we generally go for analysis of the data. So when we talk about data and variables, so the data is often discussed in terms of variables. So what is a variable? If we see the literal meaning of what a variable is, so a variable is any characteristics that varies from one members of a population to another members of the population. It is any figure or entity whose the value does not remain constant. It changes from one subject to another. A simple example may be the height in centimeters, which can vary from person to person. So basically, first of all, when we perform an experiment, we have to see what kind of data we have in our hand. So broadly, the data has been variables are classified as a numerical variable or a categorical variable. So a numerical variable is a variable where we get a number, and this is generally also known as a quantitative variable. Whereas a categorical variable, on the other hand, is a variable which can be defined by classes or categories into which an individual member falls. So if we look at this flow chart, so there are basically data types. It is broadly bifurcated as a categorical data type and a numerical data type. A categorical data type is further bifurcated as a nominal data type and an ordinal data type. And a numerical data type is bifurcated as an interval data type and a ratio data type. Now, what is the reason for such kind of a bifurcation? The reason for such kind of a bifurcation is that certain statistical methods which are applicable to an interval data type may not be applicable when we are dealing with a nominal kind of a data type. Or a statistical method that is applicable to a ratio scale data type may not be applicable when we are dealing with an ordinal scale of data type. So basically, first we have to identify what is the kind of data that we have within ourselves or at our hand, and then accordingly we can perform the different kind of categorical analysis. So if we see categorical data, the categorical data is a data that represents characteristics. So it can represent a person's gender, it can represent a person's language. So now categorical data can also take on numerical values. For example, if you're performing an experiment where we have two kinds of subjects, one of the subjects are male and the other subjects are female. So we can designate the females as 1 and we can designate the females as 0. So it is having two values, 1 and 0. But the thing we have to understand is that in a categorical variable, the numbers do not have any kind of mathematical meaning. Here we are designating the numbers simply to identify or distinguish the males from the females. to distinguish whether there is a male subject or there is a female subject, but these numbers do not have any mathematical meaning. Similarly, when we talk about nominal data we can just think of as labels. So a nominal data does not have any order. There is no change in order of its values. For example, if we see, there are two kinds of questions. We're performing a survey. The first question is whether you're married or not, and the second is what kind of languages you speak. So if we look at the first question, whether the subject is married or not, so we get a dichotomous answer. If he is married, yes, or if he is unmarried, no. So this is a data enough. So the data that we get in this case is yes or no. Or even the languages that we speak, a person can speak English, French, German, or he can speak multiple languages. So these are all data in the nominal scale, where we can just identify that it is a male or a subject. Nothing else can be done. We cannot order them. We cannot subtract them. We cannot add them. Nothing else can be done. So this is a nominal kind of a data, which is also known as a labeled. Next is the ordinal data. Now, what is an ordinal data? Our ordinal data can be thought to be slightly more updated version of a nominal data. So here also the data is categorical, but at least we can perform some kind of an ordering can be done. So in this case, if we see what is your educational background. So if he is elementary, it's 1, high school is 2, undergraduate is 3, and graduate is 4. So what is important is, you can see the difference between elementary and high school is not equal to the difference between high school and undergraduate or graduate. But what can be done is we can order it. If he is elementary, we are giving him a value of 1. If he is high school, we are giving a value of 2. So we can, depending on this, educational background, we can actually order a person's academic feature that into 1, 2, 3, 4. So because of this, the ordinal scales are usually used to measure non-numeric features like happiness, customer satisfaction, and so on. Then we have the numeric data. The numeric data is the data which is the quantitative data where we actually get values. Now again, numeric data can be divided as discrete data and continuous data. Now when we speak of discrete data, it means the data that can be counted, but it cannot be measured. That is, it will take certain discrete values. For example, if we say the number of heads in 100 coin flip, if we flip a coin once, we can either get a head or we can get a tail. If we flip a coin twice, so we can get 2 times head, we can get 1 time head, 1 time tail, and we can get 2 times tail. So there can be 4 possibilities. So here what we can do is we can actually count the number of heads in different events. So this is what is called a discrete data. On the other hand, a continuous data is a data that can't be counted, but it can be measured. For example, if we see the height of a person, certain feet, certain inches, it is basically a continuous kind of a data. Again, a continuous data can be an interval data. It can be a ratio data. And interval data, it represents ordered units that have the same differences. We saw that in ordinal data also, we could have ordered them in an ascending or a descending order. But in that case, there was no difference. But in this case, we have a true difference. So you see temperature -10, -5, 0, 5. Now the difference between 0 and -5 and the difference between 0 and 5 is same. Similarly, the difference between -5 and -10 and that between plus and 5 and plus 10 is same. So in this case, the difference also takes into account, it has got certain meanings. The problem with interval values data is that they do not have a true zero, because in this case, we are measuring the temperature of a city. And if I say a true zero, it means that there is no temperature, which is not the case, right? So with the interval data, what we can do is we can add and we can subtract, but we cannot multiply, divide, or calculate ratios, because in this case, we do not have a true zero. Now, the most updated form of data is a data in the ratio scale, which is ordered and which also has the same difference. But in this case, we also have a true zero. For example, if we talk about length, and if I say the length is 0 inches, which is a feasible case because there might not be any length. Examples are height, weight, length, etc. Now the thing is, what are the statistical methods that can be applied when we have different kind of data? For example, if we are having a nominal data, so what are the information that can be extracted out of a nominal data? First is the frequency. It is the rate at which something occurs over a period of time, or a proportion. We can find out of the total number of subjects, how many subjects are actually male and how many subjects are actually female. For example, if I'm carrying out a survey to find out how many about the number of smokers in a particular city, and I find out the female number and I find out the male number, and I can say actually say 40% of the females out of, whereas there is only 60% of the male smokers. So in case of a nominal data, since this is just a label, what we can do is we can find out frequencies, we can find out proportions, we can find out percentages. and that can be represented in the form of a pie chart, or it can be represented in the form of a bar chart. In data science, there is something called hot encoding, by which we can transform nominal data into a numeric feature. Similarly, when we have an ordinal data, we can again use the same methods that are applicable in case of a nominal data, but we can also have certain additional tools, because we can use percentiles, median, mode, and the interquartile range. Because we have said that in case of an ordinal data, what we can do is we can order the data in a particular order. Since I can order the data, so I can find out the median. For example, if it's color of different flowers, so it can be very dark red, it can be light red, it can be pink, it can be yellow. So if I order it, I can find out the median values. Similarly, in data science, by label encoding, we can transform ordinal data into a numeric feature. Similarly, when we are having a continuous data, we can use most of the methods, like we can use percentiles, medians, interquartile range, mean, mode, etc., which have been explained by the previous speakers. And regarding the visualization methods, we can draw a histogram, or we can draw a box plot, by which we can also find out the central tendency, the variability, the modality, kurtosis of a distribution. And that a histogram can't show you if you have any outlier. That is also we can use box plots. We have seen that in a histogram we can find out the kurtosis and the symmetricity of the distribution. But if there are outliers in the data, by the term outlier we mean there are certain extreme observations in the data. That is, for example, suppose we are having 10 observations and all the 10 observations are lying between 10 and 20, and there is suddenly one observation which is 200. Then that is an outlier. So if there is outlier, then that can be shown in the case of a box plot. Now, the software that I would like to speak about is the PAST software. That is a paleontological statistics software. This software is based. The advantage of this software is it is a menu-driven software. So you do not have to go for coding. Although there is also opportunity by which you can write your code in there, because there is a procedure of scripting by which you can create a script and you can write your own code. But there is also other features, like it is simple like a spreadsheet. I'll show you, and I'll also after the theoretical discussion, I'll show how to carry out certain statistical analysis in the PAST software, and a wide range of analysis can be done in this software. So this is the advantage of the PAST software. So just talking briefly about the software was originally designed as PALStat, a software package for paleontological data analysis by P.D. Ryan, D.A.T. Harper, and J.S. Whaley in 1995. Then through continuous development, PAST has increased, and now the version that we have with us is 3.22. This is a freeware which can be freely downloaded from the internet. And this is the book by Hammer and Harper in 2005, Paleontological Data Analysis, by which you can find out more details and intricacies of this software. So regarding the installation, how can you install this software? If you have Windows, you can simply search for download PAST statistical software and download this PAST3, the zipped forward, and then you can run the zipped file, and you will be done with this installation. If you are using a Mac software, then you need to download the DMG package, and you will probably have to allow the system to install the apps. Then you can go to System Preferences, Security and Privacy, and then allow apps and execute PAST.dmg, and this will you can go through with it, and this will be by which you can find out with the PAST software. So when you first open the PAST software, this is how the software actually looks. We'll also do some analysis on that, and we will see. And you can see that this is simple, just like an Excel sheet. There are rows and there are columns and there are cells where you can simply copy and paste data from the Excel sheet. And on the top, you have a file menu, you have edit menu, transform, plot, univariate, multivariate, model, diversity, time series, geometry, stratigraphy. Now, I am not talking about the script, because in this script, you can actually run your own code. So I'm not going into the script, but if you see the other the menus that we have, from these menus you can choose what kind of analysis you want to do. Take the univariate. If you want for a univariate analysis, if you want to go for an ANOVA, you will find it in the univariate. If you go for multivariate analysis, like principal component analysis or a clustering analysis, then you can find it in a multivariate kind of a tab. If you want to plot certain values, you will find it in plot. So entering the data, entering the data in PAST is very simple. You saw that PAST is in the form of rows and columns. So what you can do is you can simply type the data, or you can copy the data copy paste the data from the Excel file. So here it is. You can also write genetic sequence data coded using CAGTU. The lowercase are also accepted. And if there are missing data, you can put using a question. So if you want to select a row, you can select on the extreme leftmost column, then a row will be selected. A column can be selected on the top row. The entire column will be selected. This we will see gradually. This is important. This is the important thing is to importing data from Excel. Now we know that in most of our analysis or most of our research work, we generally keep data in an Excel file. So what we can do is we can simply copy the data from the Excel and we can simply import it in the past software. Now there are certain transform menus in the past software that we will see. Now what we can do in the transform menu is that we know that obviously there are certain situations where we need to transform our data. In many cases, the data may not follow a normal distribution, or there are certain other reasons by which we can transform our data. So what are the different type of transformations that we have? The first is a logarithmic transformation. If you have the data, you can simply select the data and go for the logarithmic transformation. We'll see in the practical sessions, by which your data will be transformed into the base 10 logarithm. We know that base 10 logarithm does not exist if the data is 0 or negative. So if the data is 0 or negative, what we need to do is we need to add a constant with the data, say 1. Similarly, we can go for subtract means. In subtract means, what we'll do is that it will subtract mean, the column mean from each of the selected columns from each of the cells. Similarly, the trend can be removed. The function removes any linear trend from a data set. This is done by subtraction of a linear regression from the y values. All the values can be converted into terms of row percentage of the row sum. We can normalize the row length, and we can also perform the famous box-Cox transformation. We know that there are certain situations. Actually, box-Cox is one of the most important family of power transformations. We know that there are certain situations where the data that we have or that we have collected might not follow a normal distribution. And we know that in statistics, whatever analysis we do, the basic assumption is that the data has to be a normally distributed data. So if the data does not have a normal distribution, so what we need to do is we can go for a box-Cox transformation, by which this transformation, the data will be distributed as a normally distributed data. So the box-Cox transformation this is the mathematical details of the box-Cox transformation, but there is a simple feature in the past software by which the box-Cox transformation can also be done. The next menu that we have in case of past is the plot menu, by which we can different kinds of graphs can be plotted in this software just by clicking. The advantage of this software is that you did not write any codes, you did not write any difficult scripts. Just by copy and pasting the data, and by clicking certain menus, you can obtain different graphs. So if you see the plot menu, the first is a graph. So the plot one or more columns is separate. The x coordinates are automatically will be set to 1, 2, 3. So there are 5 plot types available, which you can select from here. Here you have plot style, line points, or different you can select it. Then the row level option, select x-axis. There is also something. Okay. If you see here, there is something called log y. That is, if you want to transform your y ordinate into a logarithmic scale, that can also be done from here. Then there is 3-point average, there is row labels, and there is also flip axis. By flipping axis, this axis will simply get flipped. The x-axis will become the y-axis, and the y-axis will simply become the x-axis. The sequence, if there is a trend in the sequence, that can also be smoothed using a 3-point average. The next graph that we have is an XY graph. So again, here you have different plot styles. It can be a line, it can be a point. So it plots one or more pairs of columns containing an XY coordinate pair. If you have an XY coordinate pair, suppose you have the years on one side and you have the rice production on the other side, that can be plotted using an XY coordinate graph. Again, the graph can be smooth using a 3-point average. 95% concentration ellipses can also be drawn. If here you have 95% confidence ellipse, and the convex hulls, if you are performing a principal coordinate analysis or a principal component analysis, in that case you can perform a 95% confidence ellipses can also be drawn. The convex hulls can also be drawn. The minimum spanning trees can also be drawn using this graph feature. Then you have a histogram. So it plots histogram for one or more columns. The number of beans in this case is by default selected to an optimal number, where S is the sample standard deviation and IEQ is the interquartile range. But you can also change the number of beans. In this case, it is 7. You can change the start and the end. There is something called fit normal. If you check this box, it will show the normal curve, and it will show that whether the data is a normal fit or not. You can also see the kernel density and also flip the axis. Then there is a bar chart and there is a box plot chart that also can be drawn. This is how a bar chart looks, and this is how a box plot looks. Now, the difference in case of a box plot is that you find out the third quartile, you find out the median, you find out the interquartile range, and you can also find out the outliers. Then there is also options of pie chart. You know that pie chart can be drawn, but find out the different percentage or the different proportions. You can also go for a stack chart when you have more than 3 or 4 variables, and you can also go for a stack chart in the form of a percentage. This is the stacked area chart with percentage options, and the axes have been flipped. You can also draw a normal probability plot, which is also known as a QQ plot or a quantile plot for one or more columns of the data to see whether the data is actually following a normal distribution or not. You can also go for ternary plots for 3 columns of data, normally containing proportions or compositions. If you're having, say, proportions or compositions, there are different. If you're making a mixture with 3 different ingredients and there are different proportions, then you can go for a ternary plot. So this is computed using the kernel density methods. And then there is also a bubble plot. The bubble plot is actually a 3D plot. The negative values are not shown, and this is how you get the bubble plot. There is also a matrix plot. So 2-dimensional plot of data matrix that you can do using a grayscale, or you can do also use color. It includes contouring. If you have a large data matrix and the missing, you can obtain the missing matrix plot. Now there is something called mosaic plots. These are the different plots that are available in the plot menu in the part software, and you can, depending on your situation, you can choose different graphs. So this shows the proportion in a 2-way or a 3-way contingency table as area of rectangles. So here there is one of the examples. If you see, this is an example that has been taken from the Wikipedia page of a Titanic example. So if you see the number of people who were there in the Titanic, here we have sex, here we have class, here we have survived, whether yes or no. So the sex is as male and female, and whether they were passengers of the first class, of the second class, of the third class, and whether they have survived or not. And this is how the mosaic plot eventually comes out to be. You see in first class, this is the proportion of males and this is the females who have survived. Similarly in second class, third class, and then we have the crew. So if we have this kind of a data, we can generally go for a mosaic plot for a better representation of the data and a better graphical representation of the data. Then we have a radar chart. This can also be done in the software for visualizing multivariate data. Obviously, if we have multivariate data where we have more than 2 variables, one radar chart polygon will be plotted for each row in the data. So in the example, the data consists of 2 rows. You can see P34 and P26, and there are 6 columns. The grid lines can be shown as polygons. The model will also accept a single column of data. So now suppose if we have a cycle, now another use of a radar data is when we have a cyclic data or a circular data. For example, we are monitoring an animal activity for 24 hours, or we are monitoring sunlight for 12 months. So in that case, the data that we have is a cyclical activity or is a circular activity. So in that case also, we can generally go for radar charts. Then we have the 3D scatter, the bubble or the line plot. This is similar to the bubble plot that I have discussed just a few slides back, but this is a 3-dimensional representation of the same. Then we also have a 3D surface plots. So of a data matrix or elevation values. So you can see the colors are assigned according to height of the surface. So vertical exaggeration is adjustable. Here you can elevation, you can adjust the vertical exaggerate. You can adjust the style, and this is how you get a 3D surface plot. Then there is something called univariate. Now, univariate tab is a very fundamental tab by which you can calculate all the descriptive statistics at one go. You need not go for calculating the mean separately or the median separately or the variance separately. You just select the column of the data that we have, and you click on the univariate statistics. You see you get N, which is the total number of observations. Then you find the minimum value, the maximum value, the mean, the standard error, the variance, standard deviation, median, the 25th percentile, 75th percentile, skewness, kurtosis, the geometric mean, and the coefficient of variation. All these things can be calculated just by clicking one menu. And if you want to go for a bootstrap sample, you can also select a bootstrap sample, the number bootstrap, and also recompute it. So this was all about the descriptive statistics that we have. Next is we talk about inferential statistics. Now, regarding inferential statistics, the basic rules for performing any statistical test is there has to be an assumption and there has to be a hypothesis testing. Whenever we perform any experiment or we go on performing any research, there has to be a certain assumption behind it. For example, if I'm applying a certain amount of fertilizers, I would like to believe that this fertilizer will bring about a better yield as compared to the existing fertilizer. So that is my assumption, which is an assertion about the population parameter. And that is also my null hypothesis, H0. And then we will have certain hypotheses to counteract the null hypothesis, which is known as the alternative hypothesis, which may state that the new fertilizer is not better than the existing fertilizer, or it is not indifferent to the existing fertilizer. So now, depending on the situation, we may have a t-test, we may have an f-test, we may have an chi-square test. And we find out the p-value, which is the probability value, to see the significance of the statistic. Significance means whether the statistic is significantly different or there is no significant difference between the existing statistic and the new methodology. So this is the entire method of how you can actually perform an analysis. So you see, we have a null hypothesis, which is the hypothesis of no difference, or the assertion of our research work. That is, by performing this research work, I would be able to say that the new method is better than the existing method. Now, there can be two possible outcomes of the null hypothesis. The null hypothesis may be true or it may be false. Suppose the null hypothesis is true and I am accepting my test accepts it. So this is a correct situation. So in this case, the confidence level comes out to be 95%. It may so happen that our null hypothesis is true, but my test is rejecting the null hypothesis, which is an erroneous situation. It is also known as type 1 error, or the level of significance, which is usually fixed to be at 5% in case of biological experiments. For medical experiments, it is fixed at 1%. The 5% level of significance means that out of the 100 cases, there are 5 cases in which we can actually go wrong. Similarly, if the null hypothesis is false, meaning that there is no difference between the existing methodology and the new methodology, in that case, I can accept it. So even if the null hypothesis is false and I'm accepting the null hypothesis, means this brings out to be my second error, which is the type 2 error. And if it is rejecting, then actually we are performing the test is correct, and this is also known as the power of the test. So this is how we actually set up our probabilities before performing any statistical test. Now, this is a flow chart which is very important to know, like what kind of test is used in what situation. Okay, so suppose number of groups to be compared, means how many groups we are having to compare. So it can be 2 groups, it can be greater than or equal to 3 groups. If there are only 2 groups that needs to be compared, there can be unpaired observations, there can be paired observations. Now the difference between unpaired and paired is, in case of paired, the subjects are same. In case of unpaired, the subjects are different. Paired means, for example, I have 50 individuals. These 50 individuals are given the same medications and they are again reported. So in this case, the subjects are same. Again, the analysis, the test that we can perform can be parametric or it can be non-parametric. If we go for parametric, in case of this, we have unpaired t-test, for which we have to assume equal variances, which can be done using a student t-test or a Welch test. For a non-parametric method, we have to go for a Mann-Whitney U-test. If there are paired observation, again for parametric, we go for a paired t-test. For non-parametric, we can go for a Wilcoxon signed rank test. Non-parametric methodologies are generally used in those situations where we have to not idea about the parent population from which I am drawing the sample. Because in case of a parametric test, there are certain observations and certain assumptions which needs to be fulfilled. For example, I have to assume that the parent population is normally distributed. The observation drawn are random, they are independent. Whereas in case of if I have a doubt that these assumptions will be fulfilled or not, then I can go for a non-parametric assumption. Similarly, if there are greater than 3 groups, then it can be unpaired or paired. For unpaired, again parametric, if there is an equal variance, I can go for a one-way ANOVA. If there is an assumption that there is no equal variance, I can go for a Kruskal-Wallis test. Similarly, if there are paired observations, I can go for a one-way repeated measure ANOVA. For non-parametric, I can go for a Friedman test. In both of the cases, I'll see whether the probability value is less than 0.05. And if it is significant, then again, I can go for a multiple comparison or a post hoc test. Now, this flowchart is very important because this will help you to identify what is the test that you have to perform in which of the situation, which is very important, because if you're selecting the incorrect test in incorrect situation, all your analysis and all your research work will be futile. Now, you see, I talked about the 2 sample t-test. So here what we do is we compare the mean into 2 independent groups of observations using a repetitive sample. Now, what are the assumptions? The assumption is that the samples must be independent. That is, there should not be any correlation between the 2 samples. The normality A small departure from normality is not crucial and it leads to only a loss in power, but we assume that the parent population is normally distributed. And there is an assumption of homoscedasticity, which means that the variances of the two populations are equal, which can be checked by a Leibniz test. So this is an example of a situation. We consider the comparison of the mean body weights at the time of mating in one group of EVs which have been flushed, that is, which is put on a high pain of nutrition for 2-3 weeks prior to mating, and there is another group which have not. Then each EV in a random sample of 54 EVs is randomly allocated to the flushed or control group. So you see in this case there are two groups. One is control and the other is flushed, and there are total 54 EVs. And now I need to check that whether the flushed ones are better than control ones, or in other words, I can say that if there is any statistically significant difference between the control EVs and between the flushed EVs. So if I have this kind of a situation or this kind of an experiment, then what I need to do is I need to do an unpaired t test in such kind of a situation. So in this case, the null hypothesis is that the mean body weight in the population of flushed and control EVs are equal, meaning that there is no difference between the mean body weights of the flushed and the control. That is, the flushed diet does not have any impact on the EVs. So, and the alternative hypothesis is that there is a difference. So this is the t statistic, that is the difference in means by the standard error. Now, when we perform the analysis in case of a software, what we can see is, you see the group control and flushed, and then the total number of observations in the control was 30. The total number of observations in flushed was 24. Total it was 54. And this is the mean and standard deviation and the standard error of mean that we get. So this is the descriptive analysis that we have performed first before carrying out the actual test. Then I have performed a Leibniz test because I need to check whether they have got the homoscedasticity of variance or not. And you see it comes out to be significant. So the first line of the t-test result, equal variance assumption, is relevant in this instance because the result of Leibniz test indicates that the two variances are not significantly different. Now, how can we represent the values of the results in case of a scientific journal for publication? The first thing that can be done is that it can be represented in a tabular form. It can be represented in a tabular form. For example, here I have mean plus minus the standard error or the standard error with both t and p values. So in this case, you see I have represented by control and flushed. I have found out the mean plus minus standard error. I have also put the t value and I have also put the p values. Now you can skip this t value for a scientific journal and you can simply put this p value also, which brings me to my next table. I can also represent it with an asterisk so as to indicate that these are, in the first instance, I have represented using a p-value. I have given the exact value of the probabilities. Instead of giving the exact value of probability, in order to show that whether the control is significant or the flushed is significant, what I can do is I can put asterisks or I can put some superscript so as to indicate that these are significant. For here, I have put the key asterisks and I have shown that for both of the cases, the p-values are less than 0.05, indicating that the control and the flushed are significantly different from one another. Or what I can do is, I can say I have abbreviated the flushed with an superscript A and I have abbreviated the control with a superscript B. And then I have saying that means with same row carrying different superscripts A and B are significantly different because the probability values happen to be less than 0.05. So this is how I can represent my results when I am publishing it in the scientific journal. So this is the first way of representing the results in the tabular form. Next, what I can do is I can put using a graphical representation. So in a graphical representation, what I can do is I can put a simple bar diagram with different superscript as A and B, or I can represent a box plot which will again show the interquartile range and the median and the values. And to put my results in what should we do is our formal sentence must include the dependent variable as well as the independent variable. We can include the exact p-values unless the p-values is less than 0.001. The direction of the effects as evidenced by the reported means, that is the degrees of freedom, the statistical significance, these all things has to be mentioned in the scientific communication. Like for example, we can say there was a significant difference in the mean body weight between the flushed EV, which has a mean of 6.79 and a standard deviation of 1.94, and the control one with the t-value that comes out to be with the 6 degrees of freedom as 2.95 and the probability value is 0.018. Or we can say the mean body weight of the flushed EV was significantly different, with the estimated mean EV body weight in the flushed being this much and the control EVs. The significant higher mean body weight of the flushed implies an effect on metabolism and is expected to be associated with an optimal ovulation rate of 0.01, indicating that the chance of obtaining a difference in mean at least as large as 1.59 kg is only 1.8% if the null hypothesis is true. So this is how you have to present your result of your statistical analysis in a scientific journal, so that the reviewer can clearly understand what are the statistical analysis that you have done, what is the value of your test statistics, what is your p-value, which of the groups are significant, which of the groups are not, and what you want to mean by your statistical analysis, and what is your research ultimately. Then we were talking something about the pair t-test. So we say that in pair t-test, the thing is, you see, the participants has to be same. There has to be a same dependent variable, and there has to be a same condition or a treatment. You see here there are two time points, time point 1 and time point 2. In time point 1, we have certain participants A, B, C, D, E, and we have the same participants A, B, C, D at time points 2. So clearly we can see that there are same subjects in both of the two cases. Now, these are some of the methods of pairing. There can be self-pairing. Each animal is used as its own control before and after. There is natural pairing. Each pair of animal is biologically related, like litter mates. They can also be used. We can also go for artificial pairing. And the assumption is again that there has to be paired observations and there has to be normally distributed parent populations. These are the assumptions. So this is one of the experiments or the situation which shows that if this is a this kind of situation exists, ever comes to you, then you have to perform a paired t-test. See, Nelson and others in 1998 conducted a randomized crossover trial on 2 diets in 11 insulin-dependent diabetic dogs. They measured serum glucose as the variable indicating the quality of diabetic control. The diets contained either low insoluble fiber or high insoluble fiber. Now, each dog was randomly allocated to receive a particular diet first. The dogs were adapted to the diet for 2 months and then fed it for 6 months. Evaluation was performed at a 6-week interval. As a study ran over 16 months of each dog's life, we might expect changes in the animal's metabolic response. So here we can see that there are 11 insulin-dependent dogs, diabetic dogs, and these dogs are the only subjects. And these 11 dogs are given both low insoluble fiber diet as well as high insoluble fiber diet at different point intervals of time. So thus we can see that the dogs are same, the subjects are same. So this is an example of a paired t-test. So if this kind of a situation ever comes to you, so you have to go for a paired t-test. You cannot go for a unpaired t-test in case of this situation. Here you see that there are 11 dogs, and each of the dogs are given the low fiber diet as well as the high fiber diet as different time intervals. Next, we come to something which is very important called ANOVA, which means analysis of variance. Now, ANOVA is a method that is generally performed when we have more than 2 groups, when there are 3 or more groups that needs to be examined or that needs to be compared with. So analysis of variance, what we try to do in the analysis of variances, we try to partition the total variability that is present into data into known causes as well as unknown causes. The unknown causes is known as error or the residual variation, which we try to minimize, and the known causes are the factors. So the source of variation comprise of 1 or more factors, each explained by the level of categories of that factor. The 2 levels, male and female, defining factor. So we can access the contribution of the different factors to the total variation by making appropriate comparisons. The variation is expressed by its variance. Suppose, for example, we have 4 groups. Okay. In that case, we can go for an analysis of variance. So you see, in this case, this is the flowchart by which an analysis of variance actually performs. So the total variability present in the data at stage 1 is divided in between treatments and within treatments. Again, between treatments, we can have factor A variability, factor B variability, and we can also have interaction between A and B. So this is how an analysis of variance actually works. So using an analysis of variance, again, we have a broad spectrum of different experimental designs depending upon the situation. Again, we can have like the completely randomized design or the randomized block design or a Latin square design, again, depending upon the different designs that we have. One-way ANOVA is nothing but an extension of 2-sample t-test. In 2-sample t-test, we have only 2 groups. Here we have more than 2 groups. Similarly, we have a one-way repeated measures as ANOVA, which is an extension of the paired t-test. For example, when the data are longitudinal and are measured on individual at successive time points. Then we have a two-way ANOVA if we have a randomized block designs, or more complex, we can have a hierarchical structure, a nested structure, a Latin square, or a split block. So these are some of the common experimental designs that we employ in our general day-to-day life, and are very common when we have biological data or we have a biological or life science experience. So this you see is the layout of a one-way ANOVA. Here you see there are three groups. One is control, then we have diet 1 and diet 2, and the subjects. Now, the number of subjects in each of the control may not be same. So if this kind of an experimental situation you have, we can go for a one-way ANOVA. This is a one-way repeated measures ANOVA. You see there are exact number of observations, and then there is control, dose 1 and dose 2. So the same individual is given as control and also dose 1 and dose 2. And this is a two-way ANOVA, where you are having three portions and they are also having two feeds. So in this case, you see that there are two sources of variation. In this case, there are only one source of variation. That is, there is only control, dose 1, dose 2. Here also it's same, but here we have a two sources of variation. There is portions and there are also feeds. So since we have a two-way source of variation, so we have to perform a two-way ANOVA in this situation. So this is one of the experiments of a one-way ANOVA. By Nigam A.K. and Gupta V.K., a feeding trial with three feeds: pasture control, pasture and concentrates, and pasture. And minerals were conducted on a sheep farm, Mysore, to study the effect of wool yield on the sheep. For this purpose, 25 EV lamps were allotted at random to each of the three treatments, and three treatments at weight records. You see there are three feeds which were given. So I'll try to show the analysis of this after the theoretical discussion. Then we can also perform correlation and regression. We know that correlation, where we find out that whether there is a significant association between the two variables or not, and regression is the mathematical relationship between the two variables. With this, I come to an end with my theoretical presentation. Now I would like to show the past software and how to use it and how to perform some of the statistical analysis that I have discussed in my theoretical presentation. So is the software visible?
Yes, I think it's visible now.
Okay. So when you open the past software, this is how your software actually looks. And you see that there are different tabs. This is the file tab, by which you can open a new script. You can open an existing script. You can save, then save as. It can be saved in different formats. Then you have about and then exist. And this is the edit, by which you can undo, redo, cut, copy, paste. You can do different observations. This is the transform that I was talking about. You select a data and then you can perform the logarithmic transformation. You can do the subtract mean transformation. You can remove trend. You can convert to ranks. You can do the row percentage. You can do normalized length, the box box transformation. In this plot menu, you can plot different diagrams that I have already discussed. You can do a graph, the XY graph, the XY with error. Then there is univariate, the summary statistics, one sample T, two sample T, F and T, ANOVA, one-way ANOVA, correlation, intra-class correlation, normality test, then Cochrane-Hensel test, risk odds, multiple proportion. In multivariate, you can do ordination, you can do the principal component analysis, the principal coordinate analysis, correspondence analysis, canonical correspondent, discriminant, partial least square factor analysis. In clustering, you can do classical clustering, you can do neighbor joining clustering, you can do k-means clustering. In test, you can go for multivariate normality, you can do MANOVA, the multivariate analysis of variance, and also ANOCM and per MANOVA. Then there is also calibration, similarity and distance indices, and also genetic sequence statistics. In model, you can do for linear model analysis, bivariate, you can do polynomial, nonlinear fit, Lewis smoothing. In diversity analysis, you can do diversity indices, the quadratic richness, beta diversity, taxonomic distinctness, individual rarefaction, sample rarefaction, the She analysis, the diversity permutation test, time series analysis. You can go for spectral analysis, and then you can build the periodogram. You can do red fit, multi-tapper, Walsh transform, Fourier series. You can do for correlation, autocorrelation, auto-association, run test, different tests. You can go for Markov chain analysis. You can go for automatic regressive moving average analysis. And then you can go for geometric analysis like directional analysis, Nadelmass, Fourier series. Then there is something called stratigraphy, And this script is open scripting if you want to go for a coding in this. Now, suppose I have a data. For example, this is a data in the Excel file. And here you see there are two groups and there are, say, 4 variables that we have. Now, what I can do is I can simply select these 4 variables, copy, and I can paste it over here. So this is how I get the 4 variables in the past. It is as simple as that. You simply select it from the Excel software and you simply paste it. And now what we can do is, if you want to edit the name of this columns, you can select here as column attributes, and you can simply name it as. Suppose I name it this one as groups. This one, suppose I name as something, say place, whatever it is, or let it be, say, length. Let this be weight. Suppose this is as width. Okay. Now, if I want to find out this length, weight, and width, I'll select all of these observations. I'll click on univariate and I'll simply click on summary statistics. And you see, in the summary statistics, you can find out all the descriptive statistics that you have for all the 3 variables, that is length, width, and width. You can find out the n, that is total number of observations. In each of the cases, it is 20. The minimum value in each of the 3 cases, the maximum in each of the 3 cases, the sum, the mean, the standard error, the variance, the standard deviation, the median, the 25th percentile, the 75th percentile, the skewness, the kurtosis, geometric mean, and the coefficient of variation. And here you also have something called bootstrap. That is, if you want to further resample and find out more bootstrap samples for it, that you can do. Now you can simply copy this one, and the entire table can be copied into a Word file or an Excel file. And if you give for print, this can entirely be printed as it is. So this is how we find out the univariate statistics. Now suppose I want to plot this width. I want to find out the histogram of this width. So I can select the width. I can go to plot and click on histogram. So this is the histogram that has come. You see the beans has been 3. You can change or increase or decrease it. And if I click on fit normal, then I can find out the normal curve also. So this shows the normal distribution. It also shows that whether the data that we have is normally distributed or not. We can also find out the kernel density if I click on this. And if I can also flip the axis as frequency and this. So any kind of plotting can be done. Suppose if I want to plot the width and weight and width simultaneously, I can go on plot and I can select the XY graph. Now you see this is the plot style that we have obtained. This is the points. The plot style can also be changed to lines. So because this is an arbitrary data, so there is no exact relationship between the two. So this we are getting some random kind of a plot that we are getting. If I want now, in this case, the Y scale is width and the X scale is weight. If I want to find out a logarithmic of this, I can select on log Y, and this will be converted into a logarithmic scale. If you see 19.5, if I take the log of that comes out to be 1.29. So this value has been converted into logarithmic scale. So this is how these plots can be done. Any plot can be done. Suppose if I have to do a multi-stack of this, so I select length, width, and weight. I go to plot, and suppose I do a stack chart. So this gives me an example of a graph style that is a stacked bar. Here we can see this color is length, this is weight, and this is width. If I want to represent it in the form of percentages, I can simply click on the percentage, and this will be coming in the form of percentages. I can flip the axis. I can change the graph style as stacked area. Instead of the bar, this will give stacked bar. And also from graph setting, I can change the color, I can change the mean, I can change the medium, symbol sizes. These all things can be done from here. As I was talking about the transformation of the data, suppose this is the length, and suppose I want to do a logarithmic transform of this data. So I simply select the entire thing. I go to transform and click on log. So you can see that this has been converted into the logarithmic scale. Anything can be done. This is weight, for example. And suppose I want to select it into transform this into a subtract mean, where I'll subtract the column mean from each of the observation. So if I select on subtract mean, you see the column mean has been subtracted from each of the observations. Now let me show some other. So again, I have a data on 4 variables. This is a biological data on say plant height and yield. And suppose these are the 3 variables A, B, C, and D. And I want to find out the correlation between these variables. I have to do a correlation analysis. So what I can do is I can select these 4 variables. Then I go to univariate and this is the descriptive univariate statistics that can be found out for all the 4 variables. I go to univariate and I perform correlation. Now you see, this is the correlation matrix, the symmetric matrix that has been obtained, which is showing the correlation between all the 4 variables. Here we can see the correlation between A and B is 0.10, between A and C is 0.05, between A and D is 0.00, and simultaneously this is a symmetric matrix. So the entries of the matrix above the principal diagonal and below the principal diagonal will be same. Now this is the correlation that we are getting. Here we can change the correlation statistic. This is the linear correlation that we are getting, the Pearson's correlation. I can check the Spearman correlation. You can see the data will change. I can check the Spearman's R correlation. If I want Kendall's tau, I can select Kendall's tau. I can select polyserial correlation, but in this case, polyserial correlation will not be possible because in that we can do only for 2 columns. And linear correlation. And this is, if we want a correlation plot, then this can also be obtained from the correlation plot. Here you see, this is the correlation between A and A. So it is 1 exactly because this is the same variable. Similarly, the correlation between A and B, the correlation between A and C, the correlation between A and D simultaneously. And here on this, on the right, extreme right, I can see the values of the correlation. Where if it is say red in color, it shows a negative correlation, and the blue color shows a positive correlation. In this data, we do not have a negative correlation. We have all positive correlations. So this is how we can do a correlation analysis in this software. Now, okay, so suppose I want to do an analysis of variance one way and over. Okay. So here we can see that we have basically 3 kinds of treatments, 1, 2, and 3. These are the 3 kinds of treatments, and there are also missing observations. And we are measuring the yield of the mice or sheep wool yield, that the data that I have shown in the theoretical presentation. So if I want to perform an analysis of variance, what I'll do is I'll select the treatment and this entire 2 columns, and I'll go to univariate and then ANOVA. See, this is the ANOVA, analysis of variance, that we get as simple as that. Here we have the between treatments and we have the within group, and I can see the p value. So by this I can say that this is a significant value, and this is the components of variance. We can also get the Leibniz test value, which will indicate that there is a homogeneity of variance, because we know that one of the assumptions is that there has to be a homoscedasticity of variance. Variances has to be equal. So everything will be obtained over here. The Leibniz test, if you want from median, that can also be done. This is the residual plot that has been obtained for analysis of variance. And if you go for 2-case pairwise difference test, that can also be done. You can also go for the Kruskal-Wallis test. You can see the Mann-Whitney pairwise and the Dunnett's post-hoc test also. So this is how you can obtain the one-way analysis of variance, as simple as that, by simply arranging your data. Now the thing is how you have to arrange your data. You see, the arranging of data is simple. On one hand, I have the treatments, which is labeled as 111, 222, and 333, and I have the yield on the other column. So this is how I can do a one-way analysis of variance. Now let me show some other analysis that is that can be done with this software. But suppose I have a multivariate data. I have a multivariate observation. Here you can see that there are y1, y2, y3, y4, y5, y6, y7, and y8. So there are 8 variables. And suppose in this data, I want to perform a clustering algorithm. I want to do clustering of this data. So that can also be easily done in case of this software. You just simply select the data. Okay. And you go to multivariate observation. Then I go to ordination, clustering, and classical. So as you can see, this is the dendrogram of the clustering that has been done on the data. Here we have the distances, and here we have the different values, and this is showing the different level of mergers. And here is the algorithm. Now you can change the algorithm. The first algorithm by default that we have is the UPGM algorithm, advanced pair group moving average method. But you can change the algorithm. You can use single linkage. You can also use the Ward's method of centroids. Then similarity index, you have the Euclidean index, Grover index, quad, Manhattan distance. So that generally the index that we obtain is the Euclidean distance, but you can also obtain other distances. You can also use other distances. You can go for a two-way. You can also compute the bootstrap samples from this. So this is how you can also perform hierarchical clustering. You can also do non-hierarchical clustering. You can also do k-means clustering. But this is a simple clustering algorithm that I'm showing. This is how you can do the hierarchical clustering, and by also changing the algorithms. And another commonly used statistical analysis that we perform in case of biological data is the principal component analysis. Let me show the principal component analysis, how we can do, and then using this software, and then I'll give an end to my presentation. So here I have one of the data on principal component analysis. So just simply paste the data, and suppose these are the variables PP, pH, NGL, and yield, and you want to find out the principal component of these variables. What you do is you simply select these variables. And you go to multivariate, and then we go to ordination, and then we go to principal component analysis. So you see, this is the summary statistics. Matrix that we get is a variance covariance matrix. You can also change it to a correlation matrix, and because we know that the principal components can be calculated from the variance covariance matrix, or it can be calculated from the correlation matrix. So that you can change from here. So these are the eigenvalues that we get, the percentage of variation. This is the scatter plot that we have of the principal components. These are the principal component scores of the 4 principal components that we get. This is the loading plot. These are the loadings, and this is the scree plot. So this is how you can also calculate the principal component analysis. With this software, a lot can be done with this software, but due to shortage of time, it is not possible to show all the features of this software. But a lot of analysis can be done using this software. So with this, I would like to call it an end to my presentation. I would like to stop the screen now.
Yeah. Thank you, Mr. Banerjee. That was a very interesting and captivating session. And it was really important to know the different statistical tests that are being used. And it was really very informative. So I think many of the issues that we biological people face during this data analysis and all, particularly what kind of test that should be applied to biological data, you have made them clear. And thank you for joining with us. So we can move to the question answer session. And I would request all the participants, if you have any question, do put them in your comment section so that we can ask them. I think there is 1 or 2 questions, so I would like you to take. Maybe this is one question from a participant. Can you see in the screen?
Yes. So can we do t-test? Yes, we can do t-test. We can do chi-square and z-test by this software. The menu that is univariate menu, I have shown using a ANOVA. So apart from ANOVA, you will find that there are also provisions for doing a t-test. There are also provisions for doing a chi-square test. Like if there is an association, you can do the association chi-square test. You can do the chi-square test for homogeneity of variance, and you can also perform a z test. This can be done using this software. You will find all these tests in the univariate menu of the software.
There is another question over here.
Yes, regression analysis can also be done in this software. Just I showed you correlation analysis. You go to that menu, drop down menu. You will find regression analysis. The option that you go for regression analysis, you can perform the regression analysis. You can perform scree plots. You can also do scatter plots, and you can also do other kind of correlation analysis. Like the correlation analysis that I showed was the simple product movement correlation analysis, but there are also other correlations like intra-class correlation, multiple correlations. Those things can also be done in this software.
Okay, and I think there aren't many other questions, only certain comments there is one question right now.
Yes, we can heat map. I knew this software. Heat map, we can also perform heat map in this software. You can perform heat map. See, heat map is just, you can say, analysis of another way of representing the analysis of variance. The analysis of variance that we represent in the form of a tabular form. If you want to make it more captivating, you can do it a heat map. You can represent it in the form of a graphical software, showing the differences between the So heat map can also be made using the software. You can do dendrogram analysis. You can do phylogenetic analysis. Heat map can also be. I'll share one manual. You can drop me an email if you have any doubts. I can share you the manual, and I can also show you the method of how to perform a heat map analysis. I'll share one of the manual of this software with the organizers, and eventually you can get it from the organizers, where all your queries will be explained with graphical explanation of how you can do it step by step.
Yes, so that is a very good suggestion. So yes, I would request you to provide us certain tutorials or maybe manuals.
Surely I'll provide them.
Thank you, Mr. Banerjee, for joining with us for this session, and hopefully we will again, maybe some other time, we'll again join and do certain comparative sessions on R and other some other things like that can be done with R, like gene expression studies and all. So thank you for joining us again, and maybe we can end this session right now. Thank you.
Yeah. Thank you, Dr. Thakur. At the end, I would like to thank Department of Bioinformatics, North Bengal University, for giving me this opportunity of delivering the lecture. This is actually a huge topic because there are a lot of statistical analysis, and we know that biological data is really complex when it comes to analysis. So I have tried my best to show some of the methods, but there are also other methods. And I am actually delighted and profoundly grateful to Dr. Thakur and Dr. Chiranjeev sir, for giving me this opportunity. Thank you, and I'll also look forward for such opportunities in the future. Thank you. Thank you very much.
Thank you. So, thank you, Mr. Banerjee. And so we have come to the end of this 3-day training program. And I would like to thank all the participants for joining with us. Many of you are joining with us live, and many of you are viewing the program maybe later. You are caught in some other program, other work and other things. Okay, you are caught with us with the program later. So we have come to the end. One thing I would like to say to all the participants is that maybe in a day or two, we will send you the feedback form for this session. To please do fill the feedback form. It will give us an idea about what are the things that we need to improve. And you know that this online technical session is also new for us, because we are more used to giving the hands-on trainings and practical sessions. So this online training thing is a new thing for all of us. So we are in a learning process too. So we need to know our lacuna so that we can fix them. And one thing I would like to say particularly is that we intend to do many such other online programs, maybe in NGS or metagenomes and microarrays. So we are planning to do many such online programs, and in the future we may do some. So please do tune with us, and we have the email IDs of all our participants who have registered over here. So maybe whenever we are planning of a new training program, we will let you know. We'll let you we'll supply the flyers, and we'll let you know. Please do tune with us, and the only request is that please do fill the feedback form that we will provide. And regarding the PPTs and the email IDs of that, we will do that. We'll send you the PPTs in a PDF format of all the speakers and whatever the material they have, practical material they have shown. So we will share some of the material with you. And thank you all for joining with us. Hope you have enjoyed the session, and hope you have learned certain new things that you can apply in your research, in your practical field. So thank you all for joining with us. So I'm ending this session, and maybe next 2 or 3 days we'll again email you with all the feedbacks, and maybe probably we'll also send the materials in the form of slides and other material. Thank you.